Hi everyone, my name is Xiaohui. I'm from University College Dublin, Ireland. And today I'm gonna to talking about the baking releases noticeable microplastics from the polyethylene terephthalate bakeware. And first of all, I want to introduce the UCD Microplastics Research Group. This group is led by the Assistant Professor Dr. Shi, and our research interest is the methodology development for the quantifying microplastics and understand the human health impacts of the ingested microplastics. So what is the microplastics? This is the tiny plastic particles that are from the 1 micron to 5 millimeter. And this can be shared from the plastic items such as the bags, containers, auto tire, and even the clothes we wear. And it can end up in the environment, into the air, food, water, and cosmetics. So our human beings can exposure to the microplastics through the dermal contacts, through the personal care products, and by the injection through our drinking water and beverages, also by the inhalations through the air, and by the injections through the food. So as look at the figure 3, there are lots of the foods like the sugar, vegetables, seafood, meat that contain the microplastics which can be uptaken by our human body. And as our reviews, one of our reviews has shown that there are microplastics can be found in the fish around the world. And also the microplastics are found to have the health effect based on the animal and human cells experiments. And it said that the microplastics can cause the inflammations, oxidative stress, and the cellular damage to our bodies. And this also can be seen in one of our review of the potential human health impacts of the micro and nanoplastics exposure. And the microplastics even can be found in our human body, such as the lung, blood, and placentas. And cookware, it can be another route of the microplastics, because when we use the cookware during our daily life, the microplastics can be released from the cookware, and it can enter into our food and uptake by our human body. Here is one study about the Teflon microplastics released from the non-stick cookware. And in my studies, I try to investigate the microplastics released from the PET plastic oven baking pizza pans and during the baking. So my working flow is shown here. First of all, the PET bakeware was filled with the 900 millimeter water and covered by the glass lid. And we put it into the oven at 220 degrees for different heating time. And the water was collected after heating. So this water was filtered through the aluminum oxide membrane, and the particles in this membrane was characterized and quantified by the OPTIR microscope, which is the optical photothermal IR. And also we use the quantum cascade laser infrared to characterize the particles released from the PET bakeware. And here is the OPTIR and QCL IR result of the standard PET microplastics. So the spectrum of the standard PET microplastics has 4 main absorption peaks, which is in the 1725, 1341, 1263, and 1119. So the QCL-IR spectrum appears different as the OPTIR, as it has a broader absorption peak than the OPTIR. And we also do the visualizations of the bulk PET bakeware during the baking process. When I'm talking about the bulk PET bakeware, is referred to that we cut a square of the samples from the PET bakeware, and we immerse these samples into the water and put it into the ovens and heat at different times. So we use the OPTIR to detect the images features of the samples. And I can see the, oh, sorry. just exist. Sorry, I can have to start to share my screen again. And so I think this one is better. We can see the title here. So A is the mosaic optical images, and B is the visual laser images, and C is the OPDIR images at the number of the 1263, and D is at the web number of the 1725. And we try to figure out the surface difference during the heating times, and it's hard to recognize the difference of the images just by our naked eyes. So I use the GLCM to extract the image features, such as the contrast. So the contrast, it measures the local variations, and it shows that the non-heating samples has a lower value of the contrast. And another features is the homogeneous, that indicates the smooth of the images, and we find highest value in the non-heated samples, which means that the non-heated and heated surface of the samples are different, which might indicate the damage of the samples. So this image shows the magnified OPDIR images. So in here, we can clearly see that after heating, the surface of the samples become coarse, and there are some dark spots appears in the sample surface. And we also investigate the OPDIR spectra of the samples during the heating times, but we don't find the difference of the spectra during the heating. And this is the OPDIR result of the PET particles collected at the aluminum oxide membrane of the non-heated and heated samples. So A is the optical images and B is the laser images. C is the OPDIR images at the wave number of 1725. So as we can see here, that after 3 hours of the heating, there are lots of the particles released from the samples. And we also will try to investigate the spectra of the released particles, and the spectra didn't show difference during the heating. And here is the QCL results of the PET particles that collected at the aluminum oxide membrane. And similar to the OPDIR results, we found that at the 3 hours, there are lots of the particles released from the samples, and the spectrum does not show very significant difference between the different heating times. And here is the results of the size distribution of the PET particles that are collected at the membrane. And here we can see that based on the OPDIR results, we have main range of the size, which is from the 1 to 3 micron. And from the QCLIR, we didn't find the main range of the size, but it's distributed between the 3 to 336. So the difference between the OPDIR and QCLIR can be due to the different detection limits, such as the OPDIR, we have the minimum detection size, which is the 1 micron. And the QCL for these samples, we have the minimum detection limit is the 3 microns. So the results can be different between the OPDIR and QCLIR. And here is the particle numbers for that are collected at the membrane. And it is the OPDIR and QCLIR results both show that after 3 hours of the heating, there are significantly highest quantity of the particles released from the bakeware. So we recommend that the consumers, if they want to use this kind of the PET bakeware, they have better not to use it for a longer heating time. And so here comes to our conclusion is that the plastic bakeware is a potential source of the human exposure to the microplastics. And the highest quantities of the microplastics was released after the longer heating time. And also the OPTR have found that the majority of the microplastics is from 1 to 3 microns. And this size of the microplastics are found to can be uptaken by our human hair cells. So I strongly recommend that the consumers do not use these kinds of the PET bakeware, or if they really want to use it, they have better use it for shorter heating time or lower heating temperature. So that's all for my presentation. Any questions, please?
Yes, we do have time for questions. Reminder, you can type in your questions through Zoom Q&A or the Whova Q&A.
Question while we wait. I'm just wondering if you know how prevalent this type of cookware is, to try to get sort of a grasp of what the scope of the problem could potentially be. And maybe I missed it and you did talk about it.
You mean this kind of the bakeware, how it can bring? Oh yeah, because like this bakeware was shown in the Amazon, and it tells consumers that this one can be reusable. So that's it can mislead the consumer. So we try to pick out their problems and tell that you cannot use it for several times, maybe just for single use. Yes, that is the problem we are focused. Yeah.
We've got sorry, Sarah. I was just gonna say we've got a couple of questions on the Zoom. The first one says, it looked like the number of microplastics were different between the two type of lasers / imaging. Which would be more accurate to use?
Actually, we don't have the conclusion yet, because these two techniques, when we are using it, is very different. The difference can be from the detection limits, as I mentioned in my slides. And the second one is the sampling methods we use are different, because the OPTR we use less sampling area because it takes time. And for QCLIR we use more sampling area, because this one is time consuming. So that can be a second difference. And the third difference is from the background influence, because when we use the QCLIR, we use the aluminum oxide membrane as a background, and it can cause a problem. So, and the fourth one is that when we use these two kind of technologies, we use different data analyzing methods. So all of these are very different between these two methods. And for the end use, I will recommend the researchers to choose the these two kinds of technologies they want for their own studies. And still we don't have a very good conclusions which one is better for now. Yes.
Great. And then another question is, what is the maximum size limit for photothermal detection?
Maximum size. Okay. You mean the size of the particles? Here is the size of the particles is around 952 micron. That is the maximum size for these samples. And if we are talking about these technologies, I cannot record. I'm see if I can get something here. The area, the maximum area of the OPTR, you can see is a square. Is around, I'm thinking it's about 400 microns times 600 micron. That is the image you can get, if you mean that. Yes.
Yep, thank you. We are right at time.